Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I'm your host, Ben Amaralt. I'm the marketing manager at Medicinal Genomics and proud member of the CannMed team. My guest today is Dr. Hunter Land. Hunter is the Vice President of Research and Development at Biopharmaceutical Research Company, one of the few DEA Schedule I FDA-compliant cannabinoid research and development companies. He has 20 years of R&D expertise across 25 different indications, as well as 12 years of cannabinoid-focused research. Most notably, he was GW Pharma's first full-time R&D employee in the U.S., where he played a critical role in the development of Epidiolex and Sativex, and co-authored multiple protocols for the treatment of refractory epilepsy, multiple sclerosis, and pain. At CannMed 23, Hunter's presentation was titled Effect of Cannabinoids and Cannflavins on Neurodegeneration, Lifespan, and Healthspan, which is the basis of our conversation today. You can find a link to that presentation video in the show description. Topics we discussed include defining lifespan, healthspan, and longevity, why C. elegans are good organisms for studying toxicity and longevity, the entourage effect, more specifically, whether a combination of compounds has a greater effect on longevity than single compounds, how cannabis compounds compare to other longevity drugs like metformin and resveratrol, whether cannabis compounds could be used prophylactically for conditions like Alzheimer's and dementia, determining the ideal dosing range for cannabinoids, and finally, whether CBD and CBG could or should be incorporated into a healthy lifestyle to improve longevity. Before we get to my conversation with Hunter, I'd like to thank this episode's sponsor, TruLeaf. TruLeaf strives to bring you the relief that you need in a product that you can trust. Their plants are hand-grown in a facility with a controlled environment specifically designed to reduce unwanted chemicals and pests, keeping the process as natural as possible at every turn. Their mission is to provide compassionate care patients can trust when traditional medicine is not enough. Their specially trained staff works hand in hand with your physician to provide the right products at the correct dosage to ensure you get the compassionate care you need. To learn more, visit trueleaf.com. And without any further ado, please enjoy my conversation with Hunter Land. Good morning, Hunter. Thanks so much for joining us on the podcast.
Thanks for having me, Ben.
Right. Well, I'm excited to talk with you today about the potential for cannabis compounds to enhance health span and lifespan. But before we discuss the research that your team's done, I think it's important that we define those terms for the audience. So please, if you could, please do.
Sure. There's some, I would say, important intricacies, the difference between lifespan and longevity, right? So longevity is a hot topic now. Lifespan, as you would expect, typically refers to the length of life within a species. And when we talk about longevity, it's really focused on who exceeds that kind of mean lifespan throughout that in that specific species. And obviously, that's a goal of many people because longer, healthier lives is something that most of us try to achieve. Health span, on the other end, is a measure of activity and more around quality of life. So you may be able to extend life, or some people may live longer, but actually have very poor quality of life. What's the point of living longer if it's riddled with hospitalizations and lack of activity? So that could be a problem.
Great. So what made you interested in investigating how cannabis can affect longevity?
Well, this actually started out with a conversation with the FDA because, with the legalization of CBD, or the removal from the Controlled Substance Act, the FDA had a lot of concerns with, what happens if you take CBD for an extended period of time. So 20 years, 30 years, is something going to pop up down the road that could be detrimental. And it's obviously very difficult to do those studies in humans, and it's even difficult to do them in rodents. It's very expensive. It takes a extended period of time, maybe 3 to 5 years, to look at lifespan depending on that specific species. One of the, I would say, best preclinical whole organism models is the C. elegans model. It's kind of like the canary in the coal mine model. The EPA likes this for toxicity. So, for example, if there was potentially contaminated water, if they don't know what they're testing for, it's difficult to determine, what the problem is and if it's really a danger. So they may use C. elegans, and if they die early, then you probably want to stay away from that particular water. It's also been used in the International Space Station by NASA. So, what's the what happens when you're at space for an extended period of time? We don't really know what that means for humans, but we can look in other animal models. So that was really the start of this work was, hey, is this toxic, and at what level would it be toxic, and how does that, affect, the life of these in this particular model? Interestingly enough, what we found is it wasn't really that toxic until you went to super physiological amounts, and still it wasn't overtly toxic. But then we started seeing this increase in lifespan and health span. We also ran that model with cannabigerol, a lesser known cannabinoid, and we saw similar effects. So both appeared non-toxic at physiologically relevant amounts, and extended lifespan and activity levels during that life. So rather, as it would, now again, this is a model, but as it would apply to humans, it would be akin to, a 80-year-old acting more like a 60-year-old in terms of health span measures. So not only did they live longer, they were more active through that late stage of their life.
So the benefits of health span and lifespan, it sounds like it was sort of just a happy coincidence. Like you were originally looking at the toxicity, but then observed this positive effect.
Sure, we wanted, we obviously wanted to find out, how safe the compounds were, but obviously when you find out that there's a potentially larger, more profound benefit, then that's something certainly important and relevant for us within the cannabinoid industry.
Great. And so prior to this work, had any research been done into cannabis and longevity? And if so, what kind of makes this research unique?
Not to my knowledge. I think this was the first, full duration lifespan model. There've since been several other repeats where you see similar lifespan extension, some diving down into potential mechanisms. And we took those data, following these studies and said, hey, well, if this could help, general longevity, what does it then do in a diseased model, right? So then could we apply this to something like Alzheimer's dementia, or does this have broader applications to different disease states like traumatic brain injury, epilepsy, those sorts of things?
Excellent. And so, yeah, I want to talk a little bit more about C. elegans, or they're also called nematodes, correct?
Yeah, they're a nematode. Mm-hmm.
And so they have an endocannabinoid system?
They have something very akin, I would say, akin to the endocannabinoid system, but which I would say would probably be less relevant for a compound like CBD or cannabigerol, because the primary function is not through endocannabinoid system. There are some data suggesting that CBD may be a negative allosteric modulator, but in humans, it doesn't seem to. Some of the recent data presented in the last month or so, CBD may actually have like a PK/PD interaction with THC and boost exposure. So it doesn't seem that CBD, the effect at CB1 is enough to really bring down the effect of THC. So if it does work on the endocannabinoid system, I would say that's probably a minor contribution to its effects.
Okay. And so, and I think you said this too, but just so I want to make sure I understand it correctly too, that, these C. elegans and these nematodes, it's easier to study lifespan in them because their lifespan is so short, right?
Absolutely. Right.
And
16
so,
days, 20 days, so you can run a lot of different compounds in an inexpensive model. So if you wanted to look at many combinations of cannabinoids, many different cannabinoids, or like more recently, we looked at Cannflavins, you can do that kind of in parallel within the same model, and kind of get this determination of which compound is most likely to win, or at least have a better chance to win and hopefully be less toxic.
yeah, and I wanted to talk about that too, because in your presentation, you talk about sort of this initial research that you did with CBD and CBG on sort of the wild type C. elegans, and
Mhm.
saw the benefit. And then sort of, as you mentioned, did that secondary experiment on the tau model to kind of see how it could help with diseases. And what I thought was interesting about that one is that you weren't just looking at isolates there of like single compounds, but you also looked at sort of whole plant formulations and then with Cannflavins too. So kind of explain how that one differed from the first experiment you did.
Well, sure. I mean, I think for years now, there's been this interest in, rational polypharmacy as it applies to cannabinoids or the entourage effect. And I've done quite a bit of work, thanks to my mentor, Ethan Russo, years ago. Certainly had a lot of interest in this area. And, sometimes we see an entourage effect and sometimes we see what I call like a non-entourage effect. And I think this is most profound in interocular pressure models where you see reduction in interocular pressure with THC. But if you add CBD, it basically prevents that effect, that reduction in interocular pressure. So I think that's just one example of how you could see synergy or something, the opposite of synergy, right? And so this particular model, the tau model, we really wanted to dive in and see, are we seeing this additive or super additive effect with a complex botanical mixture with known components? The primary components in this botanical mixture were THC, CBG, and CBD. So, and we pegged everything at the CBD level. So we, based on the previous model, we use CBD as a positive control because we know it extended lifespan in wild type. We didn't know how it affected the tau C. elegans. Not many things are really that effective at increasing survival rate in these, highly afflicted nematodes. So we did, kind of our best guess, and then we could use that to compare to both Cannflavins and these botanical mixtures. Again, all pegged at that same CBD level. So that led us to some pretty interesting results where we do see, this profound effect with these botanical drug substances versus just the CBD alone.
Right. So score one for the entourage effect.
In this particular model, in this situation with these cannabinoids, I would say, yeah, I think it clearly shows there is synergy or an entourage effect. If you change this ratio or use different cannabinoids, I think it's important to be specific to, entourage isn't necessarily like, oh, it's a mystery soup, but it came from a plant. Well, what components are there? I mean, if I would have put CBN or CBE or some other cannabinoid in there, I have no idea if that would have helped further or if it would have hindered some of these results. And when we really didn't see with the Cannflavins. So in the second study, CBDs showed about a 10% survival rate versus 4 in the wild type at day 16. And Cannflavin was better than that at day 14. It was about 14, or day 16, it was about 14%. So, we would hope that it would have a further additive effect when you added it to the botanical drug substance. But there was no negative, but there wasn't much of an entourage, maybe a little bit of improvement, but not even to the point where, it's typically you would think of additive as 1 + 1 = 2 in terms of, I'm taking a medication for, let's say, pain, and you've got two different components. They both work on their own. You add them together, 1 + 1 = 2. Super additive would be 1 + 1 = 4. And that's what you would hope for. And that's what we saw with the botanical drug substance. But we didn't see that when we added in the Cannflavins. It was somewhat better, but not we didn't go from 25% survival to 40% survival, which would have been great, but there may be limits to how far you can push this kind of entourage effect.
Right. And so this botanical drug formulation now, is that a whole plant extract or is that just combining different isolates into like a formulation?
So, under the botanical drug guidance, you can essentially blend to a specification from extracts, and that's probably the best way to keep things consistent. That's what we did with Sativex at GW. So it was a high THC chemovore and a high CBD chemovore that were, climate controlled and consistent. But then you blend to a certain specification, so you always have that particular ratio. And essentially, that's what we did here. Is we use different extracts to blend to a specific ratio. And then we also use the Cannflavin extract to blend into this for this particular study.
Yeah, because the reason I ask is if it's, sort of a whole plant extract, is there the possibility too that there is a minor cannabinoid or some other minor compound that's kind of doing some heavy lifting that we're just not seeing?
Could absolutely be there. Don't know exactly what it would be, and again, with every plant being so different, you'd love to find this, highly potent miracle molecule that's in there, which may or may not exist. But it would have been interesting to run it with, three isolates in that same ratio and see if we get, this difference because of these minor components. There have been a couple studies that suggest that the mixtures may increase bioavailability a bit, and that may be because some of these other compounds are inhibiting CYP enzymes. So it could be, direct receptor interaction, or it could be, a pharmacokinetic interaction that may add to the benefit of a botanical drug substance.
So how do these results compare to other drugs or compounds that have been studied for this application?
There really haven't been many in the tau model. We see this is fairly, the early studies with CBD and CBG are fairly consistent, maybe not quite as beneficial as metformin. For those of you who don't know, metformin's a drug commonly prescribed for metabolic syndromes like type 2 diabetes, but there's a thought that it may extend life, and there's a couple different ways it may do that, but one of the ways is through this methionine and folate synthesis. So it seems to inhibit those two compounds, which apparently is important for life extension. Ironically, those are two known, or at least methionine synthesis and folate 1-carbon metabolism are two mechanisms known for CBD. So there may be a mechanistic overlap with metformin. And again, both compounds extended life. Resveratrol is another compound. It's a silbenoid. So has some similar components as cannabinoids in terms of like the terpene structure within that compound. Also seen to extend life. Not as applicable in humans because of metabolism in the gut. So that's something, we have to be careful with these models. Just because it extends life in C. elegans, what happens when you put it in a system like humans? What happens with metabolism in the gut and bioavailability? So all critical considerations to take into account. But generally speaking, it looks as good as many of the other compounds that we think may extend life, at least in this model.
Excellent. And so what is the next step? I mean, if you start with C. elegans, do you kind of move up the chain of complexity there and kind of test it?
Well, we could certainly look at different compounds, but I think different combinations and different models of C. elegans. But given that tau appears to be a piece of the underlying etiology of many different diseases, it may be important to look at some of these. So, for example, maybe in Alzheimer's dementia, maybe you look at rodent models or human models because we saw some effect here. Maybe this is one of the underlying mechanisms for traumatic brain injury or stroke, or a number of other things. Even cancer seems to have this. Tau seems to be important, and having these tau tangles seem to affect cellular energetics and longevity on the cellular level. So preventing those or acting as a protectant could be critical in a variety of disease states.
Yeah, and I guess that brings up a good point too. So are we saying that these compounds would be beneficial in preventing these diseases from occurring or treating them in folks that already have them? Or both?
Well, I think that's an excellent point. The truth is, we don't know. Obviously, prevention would be ideal, although generally speaking, our healthcare system's not uh, entirely focused on prevention. But what we could potentially do is prevent worsening. Right? If you let's say it does, let's say we do find out that it is genuinely protective and helpful in something like Alzheimer's. Um, can we give it early onset? What about other conditions like MS or Parkinson's that you have this neurodegenerative kind of disease? Um, can we give it early and prevent progression? Which again would be certainly beneficial to that population, and maybe not a cure, but it could help until something else is potentially better that is a cure arrives.
Yeah, and another thing that you had mentioned too is that sort of the results you were seeing was equivalent to like an 80-year-old human feeling like a 60-year-old. So be better
Base
to
activity
take that
level.
early before you are 80, or can an 80-year-old, take it
Yeah.
now and feel younger?
Well, even how well that would translate, we're not really sure, but based on the data, it would seem that taking for a period of time, probably similar to exercising your whole life or dieting your whole life, may be of benefit. To what degree that benefit exists in humans, I think is unknown, but it's interesting nonetheless.
And it must be so difficult to determine that in a human too, because there's just so many different factors that go in.
Oh, the variables, these
Yeah.
epidemiological studies around diet and vitamins and nutrition, just incredibly difficult to do. So many confounding factors that make that data very difficult to determine. But I think the key point for the early studies is that it doesn't appear to be overtly toxic at these levels. We do know in humans that there are some potential dangers around liver toxicity with high doses of CBD, especially with certain drugs like valproic acid, which is also a known liver toxin. So, I wouldn't say that high doses for your whole life are safe. There could be some complications there, but at low doses and medium doses, it doesn't appear to be a problem based on exclusively on this model. But it's a big leap to say the same for humans. But so far so good. If it looked toxic, then there would be a lot more alarm bells.
I know it is difficult to make that leap, even though I keep trying to make you I keep trying to make you do it. Um, and looking at the results, and you talked about dosing, it seemed like there was sort of a sweet spot range for the dose that really got the best effect. It wasn't necessarily the highest dose that got the best effect. So I was wondering if you could speak a bit to that.
Sure. I mean, this is we saw it across both studies in botanical drug extract and in an isolate and tau model and not in tau model, and it's not really surprising because we do expect just about everything at a certain level to have toxicity, and what we see actually CBG appears to be less toxic in this model than CBD milligram per milligram, but again, you can there's an LD50 for water, so as you go up, eventually something's going to get toxic. But I would also say, in terms of efficacy, this is it's been a theory for some time about this kind of inverted U or kind of biphasic effects with cannabinoids. I think they're more profound with THC. I think there's a narrow dose window for THC. We know that low amounts or moderate amounts increase appetite. They may help with anxiety. They may help with sleep. But when you start giving really high doses of THC, you get massive paranoia, nausea, and vomiting. I mean, it's not pretty. There are a bunch of hospitalizations for people that didn't know how much was in their brownie. So as with these sorts of physiological effects, you may see the same thing in lifespan. Taking too much may be a hindrance, or may be neutral in terms of being of benefit. We still don't know kind of where that sweet spot would be, especially as it relates to CBD and CBG in humans.
Okay. So can't necessarily apply the dosage that was effective in C. elegans to humans. Like, is there like a conversion there that you could take with in terms of what the dose would be?
Yeah, there is a conversion. It's kind of complicated how you do it in C. elegans. There's two ways. You could actually look at the exposure. You could spin them down and look at their exposure, or you could use it as a correlate with other compounds that have similar lipophilicity. So it is complicated, but that 40 nanograms per mil, or 40 nanograms, 40 micromolar, excuse me, is certainly a physiologically relevant amount, and not the same as, if I was doing work in a dish, right? Because they're basically eating the medium, so that's the concentration in their food essentially. So not necessarily their exposure on the cell level. So when you see cancer studies, oftentimes we're applying that, and that would be the cell exposure. This is kind of the food exposure, which is relevant to their internal exposure after metabolism.
Okay. And apologize if you already answered this question,
No.
but um, so what do we think is going on here? Like, what mechanism is being applied to kind of improve the health span and lifespan of these C. elegans? Do we really know that yet, or?
I don't think we know. I mean, there's CBD and CBG are, I would say, promiscuous molecules in terms of mechanism. Unlike a lot of new chemical entities and synthesized drugs, they're not really designed to work on one receptor type. They're not highly selective for one thing. I mean, we know that CBD has at least 68 known targets, and none of them are really potent. THC is certainly more clear that it works primarily as a partial agonist at CB1, but also has some other quote unquote off target effects. Know even less about CBG. I think it's difficult to determine. It may be relevant to cellular energetics or mitochondria. We know there's some um, potential mechanisms there. There's nucleotide receptors that may be important for transcription, translation. So, truth is, we don't know. There's tons of possibilities. So many that are countless. I could probably spend the rest of my life trying to determine which combination of 100 different mechanisms
Right.
are contributing, but something's going on, and how can we apply it to potentially make a medicine to help people is would be great to know. I think that's the next step.
Right. Well, good job, security. You got plenty of things to investigate.
Right. Probably work on this the rest of my life, and might, who knows.
Excellent. All right. So I'm gonna do it again, and I'm sorry, but what is so? I'm an otherwise healthy person. I exercise, I eat right, I try to take my vitamins and things like that. So, should I incorporate CBD, CBG into my routine to potentially help my lifespan and health span? Is it too early to tell, or
I think it's still too early. I mean, you certainly could do that. Things I would watch out for are things like drug interactions, and certain people with certain conditions, it may not be right for that individual. And then also just knowing where to get a quality thing that is labeled appropriately. Also, it can be expensive. I mean, this is kind of diverging a little bit from this conversation, but these are fairly high milligram doses in humans. So, taking 10 milligrams of CBD in a gummy um, may not be doing much, if anything. And if you're willing to spend on, 100 milligrams or hundreds of milligrams per day, it can get expensive. I mean, that could be, $300-400 a month. And then where do you get it? Which company is the most reputable that has CBD and CBG and an idealistic ratio for health? And I think that remains to be a kind of industry wide problem.
wow, is that the dosing level that we're talking about here? Like up to
Yeah, it
100
would be
milligrams?
akin to hundreds of milligrams in
Wow.
humans.
Yeah.
Mhm. So not, but who knows? I mean, we did see effects at lower doses, and but yes, it's probably not just 25 milligrams a day of CBD. Could be, but we don't really know.
Interesting. All right. Well, I'll hold off then.
Yeah. may be advisable. If nothing else, it'll probably save you some money.
Excellent. All right, Hunter, before I let you go, I want to give you a chance to share any other resources that might be out there that folks could read up on if they're interested in learning more about this topic. And then please also plug any ways that people can get in touch with you and learn more about the work that you're doing.
Well, unfortunately, there aren't a lot of resources in this area other than maybe PubMed or Google Scholar. We're working to make more resources and, publish these data, and we'll see how it gets out to the general public. Certainly, you could attend CannMed next year, and maybe there'll be some more great presentations on these topics. And we'll just keep going. And if there are any questions, I'm on LinkedIn, pretty easy to find. There aren't too many Hunter lands out there, so feel free to reach out there, and we'll keep you posted on. We should have some more data next year.
Excellent. Well, we look forward to that data, and I will make sure to put a link to your presentation at CannMed 23 this year in the show description so people can check that out.
Great. Thanks so much, Ben.
All right, Hunter. Thanks again, and we'll see you down in CannMed next year.
Okay, great. Thank you.
I hope you enjoyed my conversation with Hunter Land. Check out the links in the show description to learn more about the topics we discussed, and thanks again to this episode's sponsor, TrueLeaf. Our next episode drops July 19th, that's 2 weeks from today. In the meantime, I invite you to check out CannMedEvents.com and all the resources that we have available there. Most notably, our CannMed Archive, a searchable database of all the video recordings of the presentations that have been delivered at our CannMed events. There you can also sign up for email alerts so you can be among the first to get the news when we announce our plans for CannMed 24. We also hope you'll join us on social media. We're on Instagram, Twitter, LinkedIn, and Facebook. Just search for CannMed Events. And also, if you're on social media, please join our CannMed community group on Facebook. There you can connect with other members of the CannMed community, share relevant links, videos, memes, really anything CannMed and cannabis related. All right, that's it from us. Stay safe, stay healthy, and be sure to join us for the next CannMed Coffee Talk.